Hi, everyone, and welcome to the second part of the Chapter 2 lecture on the chemistry of life. In the first part of this lecture, we talked about essential chemistry principles, which led up to a discussion of why water is an essential chemical for living organisms. But that was only one of the two questions that I said we were going to talk about in this chapter. And the other one that we have not addressed yet is what are living things made of on a molecular level? So we're going to get at that question in this second part of the lecture, and you may remember that the sorts of molecules that are the scaffolds of living organisms are the sorts of things that we see on a nutrition label and that are found in our food. And these molecules are divided into four different categories. Those four categories are carbohydrates, lipids, proteins, and nucleic acids, three of which you can see on your nutrition label in the form of fats or lipids, carbohydrates, and then finally proteins. All four of them are referred to as macromolecules because on the scale of molecules, they are considered to be very large. So, in this lecture, what we're going to be doing is looking at each of these four categories of biological macromolecules one by one, and we're going to talk about how they are structured and what sort of role they play inside of living organisms. And as we do this, we're going to be cognizant of the fact that all these macromolecules are made up of, as we saw before, smaller molecular units. So all of these macromolecules are made of what we refer to as monomers. A monomer is defined as a single molecule that can be combined with other molecules like it in order to create these larger macromolecular structures. The term monomer makes sense to us because the prefix mono means one. So a monomer is a single molecule, a single molecular building block that can be combined with other building blocks to make polymers. Most macromolecules are considered polymers, which are chains of monomers put together. The prefix poly means multiple, and so therefore a polymer is a chain of multiple monomers that are put together. Now, the building blocks of every type of macromolecule look different from each other, by which I mean the building blocks of a carbohydrate are different from the building blocks of a protein, for example. However, they do have some things in common with each other. And one of the things that they have in common is the way that they get bonded together in order to make these larger molecular structures. There is a specific type of chemical reaction called a dehydration synthesis reaction, which occurs in order to link the individual monomers together to form the larger polymers or macromolecules. While every macromolecule's monomers are structured differently, as I mentioned, they do have in common that they have specific types of functional groups on them that are made of specific types of atoms. So we can see one monomer represented over here as this yellow block, and we can see on the right-hand side of it there is an oxygen and a hydrogen sticking off of it. On this one over here, we can see that it is identical to the one on the left, and on the left-hand side there is a hydrogen here. A dehydration synthesis reaction will take these two building blocks and it will put them together, combine them into one by joining them. And as it does so, it will kick out a molecule of water that is formed in the process, which is why it's called a dehydration synthesis reaction. Dehydration means loss of water, and that's exactly what goes on in this type of reaction that joins together these two monomers. So let's take a look at an animation of this reaction on the next slide here. The monomers come together. The two hydrogens and oxygen are bonded to form a water molecule, which leaves, and when that water molecule leaves, it seals together a bond between those adjacent monomers, and now they've linked together into a small polymer. So let's think about when this sort of reaction might happen in our own bodies. Let's say you've just had a workout and your body is working to build muscle tissue. It will take the individual monomers, the building blocks of proteins, and put them together to make the sorts of proteins that are necessary to build muscle tissues. Of course, the opposite happens as well, right? Living organisms not only build up large molecules, but they also need to break down large molecules. Obviously, this happens, for example, when you are digesting your food. You ingest large molecules like proteins and carbohydrates, and your body needs to break them down in order to obtain energy and nutrition from them. So, the opposite sort of reaction to a dehydration reaction is called a hydrolysis reaction. Hydrolysis reaction is named for the fact that it again involves water. Hydro means water, and the term lysis literally means to split or to break apart. So hydrolysis means to break apart the monomers by adding a water molecule that splits their bond between them. And as we will see on the next slide in the animation, the hydrolysis reaction is precisely the opposite of the dehydration reaction. The water molecule will come and break the bond between the two monomers, and then the monomers will be split apart. So that should provide you some solid background on how each of these four classes of macromolecules gets formed. And we're going to see these principles of dehydration synthesis reactions occur over and over again with each of these four categories as we walk through them. So let's get started with our first category of macromolecule. We're going to start with carbohydrates. So carbohydrates are large molecules that are made up of smaller building blocks called monosaccharides, which in colloquial everyday language are also referred to as sugars. So actually, carbohydrates are very large molecules that are made up of molecules that would be considered sugars. There are too many different types of monosaccharides to name, but you can see a few examples here on the slide. We've got glucose, galactose, and fructose, and you may be able to see that they all have in common that their names end in ose. So you can tell if something is a sugar if the name of it ends in OSE. Glucose, galactose, fructose are just a few examples. You've also got sucrose, maltose, lactose. All of these things are sugars and building blocks of carbohydrates. Let's take a closer look at one specific building block example that we saw on the last slide, and that is glucose. We are going to walk through how individual building blocks of glucose come together to form a larger carbohydrate macromolecule. So here we have two separate molecules of glucose, and we are going to see how they can come together through a dehydration reaction that chemically bonds them. I'm going to highlight the atoms that are specifically involved in this chemical reaction. On the left-hand glucose, we can see that I've highlighted an oxygen and a hydrogen atom. And on the right-hand glucose, we can see that I've highlighted just a hydrogen. What will happen is these three atoms, the two hydrogens and one oxygen, H2O in other words, will come together and join to become a water molecule that is separate from either of the two glucoses. This, of course, leaves behind the atoms that they were previously bonded to, this carbon on the left and this oxygen on the right. And these two atoms now have the need for something else to bond to. And so they will turn to each other and form a new chemical bond between them, which results in the connection of the two glucose molecules through this dehydration synthesis reaction. Now, a single dehydration synthesis reaction like this is not enough to make an entire carbohydrate. What has just been made here is something called a disaccharide, which is just two monosaccharides put together. But when you repeat this process over and over again, you form a long chain of monosaccharides, which eventually ends up turning into a carbohydrate once it gets long enough. So what is the biological role of carbohydrates in living organisms? Carbohydrates are a pretty versatile type of molecule that can be used for multiple different purposes depending upon the type of carbohydrate and depending upon the living organism. You probably already know that carbohydrates carry a lot of energy, and so in humans, for example, they are used for energy storage in the form of glycogen in the human liver. In plants, carbohydrates can be used for a different purpose. The cell walls, or the barriers that exist around the outside of plant cells, are composed of a type of carbohydrate called cellulose. So you can see that they are versatile molecules that can be used for either structure or energy storage. This brings us to our first checkpoint of the lecture, where I want you to tell me how many water molecules would be generated from the dehydration reactions required to create a carbohydrate chain made of 5 monosaccharides. Now, it may be helpful to draw this out, just very roughly sketch it without any level of detail, to see how many new bonds and how many chemical reactions would need to happen to link 5 monomers together. And I'll give you a hint. It is not 5 water molecules. So this completes our discussion about carbohydrates and means that we can move on to the next class of macromolecules, which is the lipids. Now, lipids, as we mentioned, are also known as fats, and on a molecular level, these are composed of different sorts of building blocks than a carbohydrate. Instead of being composed of something like a monosaccharide, lipids are composed of 2 different building blocks. They're made of a molecule of glycerol, which you can see on the left-hand side here, connected to multiple fatty acid chains, which is what you can see on the right-hand side. So every lipid contains a single glycerol connected to multiple fatty acid chains. And as you can see, a fatty acid chain is essentially a long chain of carbon atoms that is attached to a bunch of hydrogen atoms. And the length of the fatty acid chain can vary. This particular fatty acid contains 11 carbon atoms in its chain, and it is called lauric acid. This is the fatty acid that is commonly found in the fat that makes up coconut oil. If you had a different length of the carbon chain in the fatty acid, then you would get a different sort of oil. So when we talked about carbohydrates, we looked at how 2 separate glucose molecules could come together in a dehydration reaction to form the beginnings of a carbohydrate chain. We're going to do the same thing here with lipids. We're going to look at the making of a type of lipid called a triglyceride, which is the common type of lipid that is found in your body that stores fat. On the left-hand side here, we can see a molecule of glycerol that I have sort of lengthened out here for our purposes. And on the right-hand side, we can see 3 separate molecules of fatty acid. Each of these 3 fatty acid chains is going to become attached to the glycerol on the left-hand side via a separate dehydration reaction. So 3 dehydration reactions, 1 for each fatty acid chain. Again, I'm going to highlight the atoms that are involved in these reactions. So within the glycerol, we have 3 hydrogen atoms, and within the fatty acid chains, we have combinations of hydrogen and oxygen. Overall, each of these sets of atoms will come together and form a separate molecule of H2O. There's your dehydration. And this leaves the glycerol and the fatty acids open and available to form new bonds to each other to fill the place of what they've lost. Once that is complete, here is what your triglyceride looks like. Again, we have a single glycerol molecule on the left-hand side that is attached to 3 fatty acid chains. So we just saw how a type of common lipid called a triglyceride was formed, but there's another type of specialized lipid that we need to discuss again, and that is a phospholipid. Now, we briefly mentioned phospholipids previously in our lecture when we were discussing water and the hydrophobic effect. You may remember that from our last lecture. But a phospholipid, the structure of it is similar to a regular lipid, but the exception is that instead of having 3 fatty acid chains, it has only 2 fatty acid chains attached to the glycerol molecule here. And in place of the third fatty acid chain, it instead has what's called a phosphate group here. So we can see the phosphate group on the left-hand side docked into this glycerol molecule at the station where the third fatty acid chain would normally exist in a triglyceride, but it's not there. Now, phospholipids, as we mentioned before, are the fundamental unit that comprises the membranes of cells. And so the biological role of lipids is again complex because while regular lipids are used for long-term energy storage in the form of fats, phospholipids are equally important because they are a type of lipid that is the main structural unit in cell membranes, as we can see on the right-hand side here. So at this point, I have one more checkpoint for you about dehydration reactions and water molecules, which is how many water molecules would be generated from the number of dehydration reactions required to create 2 complete triglyceride lipids. Now, before we move on from lipids entirely and on to the next category of macromolecule, there's a few other things that I wanted to mention to you guys because these are relevant things that you hear about in relation to fats. One of them is saturated versus unsaturated fats, 2 different categories of fats that you may see on your nutrition label. The difference between a saturated and unsaturated fat comes down to whether or not the fatty acid chains contained within that fat molecule have all single bonds between their carbon atoms or if they have at least one double bond. A saturated fat contains fatty acids that only have single bonds between all of their carbon atoms, as you see on the top here. This means that every single carbon atom is in a covalent bond where it shares only a single pair of electrons with the next carbon atom. An unsaturated fatty acid, on the other hand, has at least one double bond, meaning that at least one pair of carbon atoms in the chain of the fatty acid shares 2 pairs of electrons with its neighbor. Saturation refers to the level of hydrogen that fits in the fatty acid chain, which depends upon whether there are single bonds or double bonds. As you can see in the upper fatty acid here, this fatty acid is saturated, or in other words, completely full of the maximum number of hydrogen atoms that it could possibly contain. The unsaturated fatty acid is unsaturated in the sense that it is not completely full of the maximum number of hydrogen atoms that it could contain, because these 2 carbon atoms, instead of sharing electrons with another hydrogen atom, have chosen to share electrons with each other instead. So that's the significance of saturation versus unsaturation. Now, what does that mean to you and your diet? Well, research continues to investigate the difference between saturated and unsaturated fats on our health, but the current body of evidence seems to indicate that unsaturated fats are healthier than saturated fats because saturated fats are capable of raising your cholesterol levels in an unhealthy way. And an easy way to tell the difference between saturated and unsaturated fats is based upon what state they are in at room temperature. Saturated fats tend to be solid at room temperature, so this includes things like butter or lard or coconut butter. Unsaturated fats are things like oils, like olive oil, vegetable oil, canola oil. These at room temperature will be liquid instead of solid. So that's how you can tell the difference. So we're now moving on from lipids to our next class of macromolecules, which is going to be the nucleic acids. This, remember, is the one that you don't find on your nutrition label because it is genetic material, DNA. The nucleic acids are composed of a separate type of building block that is not the building block that makes up carbohydrates, nor is it the building block that makes up lipids. It's a building block called a nucleotide. A nucleotide is a very complex building block because within it there are actually 3 different components. Overall, together, all 3 of these things together are a nucleotide. But within them, we can identify 3 separate pieces. There is a sugar, which I've highlighted here in green. There is a phosphate group, highlighted here. And then finally, something called a nitrogenous base, highlighted in the upper right. So how do these individual building blocks come together to form the larger macromolecule? That's what we're going to see next. Here we have 2 separate building blocks that have not yet conjoined to each other. I'm going to highlight the atoms once again that are going to be involved in the dehydration reaction that will connect the 2 in order to create a strand of nucleic acid. On the upper nucleotide, we can see that I've highlighted a hydrogen that is part of the sugar component of that nucleotide. On the lower nucleotide, we can see that I have highlighted an oxygen and a hydrogen together that is part of the phosphate group on this nucleotide. These are the 3 atoms, the 2 hydrogens and the oxygen, that will come together and leave their partners in order to form a separate water molecule, which makes it possible for the 2 nucleotides to then form a new bond to each other. And now we've linked the 2 nucleotides through a dehydration reaction. Now, once again, if we just link 2 nucleotides, that doesn't really form a strand of DNA. It's when this process happens over and over again that you get long strands of nucleotides that then form a nucleic acid, which can be either a DNA or RNA molecule. And finally, what is the biological role of nucleic acids? Well, just think about what you already know that DNA does for you. Nucleic acids, like DNA, store and transmit genetic information. We'll talk a lot more about this in future chapters, but for right now, that's all you need to know about nucleic acids. So this brings us now to our final category of macromolecules that we have yet to talk about, and that is the proteins. We saved the best for last because the proteins require a little bit more discussion than the other classes of macromolecules. But we'll start in the same place as we did with the other 3 classes by talking about what the monomers of proteins are. The monomers of proteins are called amino acids, and you can see a representation of what an amino acid looks like on a molecular level on the left-hand side of this slide. An amino acid, sort of like a nucleotide, has a complex structure with multiple internal parts that we can identify. Every amino acid contains a central carbon atom, and we can see that central carbon atom right here, that is bonded to 4 different things. One is an amino group. You can see that on the left-hand side. That's part of the namesake of the amino acid. Next, a carboxylic acid group on the right-hand side. And again, you can see that that's part of the namesake of amino acid as well. Up top, we have a hydrogen atom. And then down below, we have something called a quote-unquote R group. And that R group actually can vary in its chemical structure. It can be many different things. For example, in the amino acid known as glycine, the R group is simply a single hydrogen atom. In the amino acid known as aspartic acid, the R group is much more complex. In the amino acid known as serine, it's different still. And in fact, there are 20 different types of R groups that are found on amino acids, and all of them are displayed here for you to see. Now this chart has nicely divided the amino acids into different categories based upon the chemical nature of the structure of their R group. All of the amino acids that are listed in this upper category right here, all of these amino acids have a non-polar R group, meaning that within their R group, the electrons are shared equally across the atoms that are engaging in covalent sharing bonds. Down below the nonpolar group, we can see the polar amino acids. And you may remember that polar is the opposite of nonpolar. What this means is that the atoms in the R group of these amino acids are not sharing their electrons equally between them, and so they have these dipoles that have a slight positive or negative charge to them. And then finally, at the bottom, we have a category that is a full-blown electrically charged amino acid, meaning that it's not just sharing electrons unequally with a partner, it has either lost or gained electrons entirely. On the left-hand side, we have two amino acids that have an overall negative charge because they have gained an electron. And although it's hard to see, that negative charge is localized on these oxygen atoms right here. You can see the little tick mark in the upper right of that oxygen indicating that it has a negative charge associated with it because it gained an electron. Over here, we have three amino acids that have the opposite of that. They have a positive charge because they have lost an electron. And again, although it's hard to see, there's a little plus sign localized around these nitrogen and hydrogen clusters here, indicating that there is a positive electrical charge on these three amino acids. Now, based upon what we learned in our previous lecture, I want you to tell me which amino acid would be able to dissolve in water, asparagine or tryptophan, and why. So you'll have to find these two amino acids and which category they belong to in this table. And then based upon what you know about nonpolar and polar things and their capability to dissolve in water, which one of the two could dissolve in water and why. After that, I want you to tell me a pair of amino acids that would exhibit an electrical attraction to each other. So just as we've done with the previous categories of macromolecules, we're going to take a look at how a dehydration reaction mediates the bonding of two separate amino acids in order to start forming a macromolecule. So here I have two generic amino acids here on the slide. They're separate from each other, and we can see that the R group is simply represented with the letter R, indicating that it can be variable. They're not a specific amino acid. Again, I will highlight the atoms that are involved in this reaction. I'm picking up the OH from the left hand and the H from the right hand amino acid. These three are the ones that will get together and form a molecule of H2O, leaving behind the opening for the left-hand amino acid to bond to the right-hand one, and for the two to come together to form a 2-unit molecule here. Now this in itself, again, is not a protein, but when you do this over and over again and get a long chain of these monomers together, that's when you have a protein on your hands. And here's what such a protein might look like with various side chains filled in instead of the generic R groups. Now, the thing about proteins that makes them a little bit more complex than other types of macromolecules is they don't sit nicely as chains like this. And in fact, they have this complex folding behavior that allows them to take on a three-dimensional shape. The chain that you see right here might look something like the chain that you see on the upper part of this slide, but it won't remain like this. And instead, it will form patterns and three-dimensional shapes which fold on each other in various ways that allow it to assume the complex structure of the protein. So why are proteins important? What is their biological role? It turns out that proteins are the most versatile of the 4 classes of biological macromolecules because their role can be many different things. It can be structural. For example, collagen is the protein that composes the structure of your tendons and connective tissue. Keratin is another protein that composes the structure of your hair, and that's what you see in the upper right of this slide. They can also be functional, meaning that they can carry out a specific function within an organism. For example, hemoglobin is a very important protein that is responsible for carrying oxygen within your red blood cells through your bloodstream and to your organs and tissues. And then lastly, proteins can be catalytic. Most enzymes are made of protein. And what does that mean? What is an enzyme and what does it mean for them to be catalytic? Well, a catalyst is something that facilitates and speeds up a chemical reaction. And this is important because there are literally millions of chemical reactions happening in your body at every single second. Virtually every chemical reaction in every living organism is catalyzed or sped up by a specific protein enzyme. What enzymes do is they take molecules and they either break them down or they build them up. Here in this animation, what we see is an enzyme taking a molecule of sucrose, a type of sugar, and performing a hydrolysis reaction on it in order to split it in half into two molecules of smaller sugars. So this is a reaction that happens inside of our bodies all the time when we are digesting, for example. However, without the help of an enzyme, it would take much, much longer for this chemical reaction to occur. With the help of enzymes, it can occur at a reasonable pace that allows a living organism to digest and survive. So enzymes make life possible by speeding up all of the reactions that are involved in keeping organisms alive. We are going to round out this lecture with a few checkpoints to help us review everything that we've learned so far. First of all, here I want you to match the biological macromolecule with the name of the building block or the monomer that composes it. And finally, match the macromolecule with the function that it performs in a biological organism. And once that is complete, you are finished with this lecture.